Welcome to Quran Academy. Today we will be discussing photosynthesis. So here is the general equation. We will get more into depth about this. So there are three phases of photosynthesis. The first one being light reactions. They are dependent on light to function. So that takes place in the chloroplasts. Water is entered and protons exit, as well as oxygen exits. Secondly, ADP and ATP are interchanged with the addition of energy, and NADPH is reduced to NAPH, and vice versa. And third is the Calvin cycle, with carbon dioxide entering and glucose exiting to either go to the roots or to be stored as starch. Now I will talk about light reactions. So here I am drawing 4 proteins that are on the outer membrane of the chloroplasts. The first one is called photosystem 2, and it has a chlorophyll pigment called P680, and underneath is a Z enzyme, and I will discuss the function later. So the second protein is called photosystem 1 and has a pigment of P700. The point of the arrow I drew shows the path that the electrons follow. Thirdly is the NADP reductase. So the photosystem 2 and photosystem 1 must get light to excite the electrons to be passed in the path shown, and the Z enzyme splits water into positively charged hydrogen and oxygen. Stroma is the outside and lumen is the inside, or called plumen, and the point is to get a surplus of H pluses inside the plumen. Lastly is the ATP synthase, where 2 ADPs become 2 ATPs. Now I am drawing the phospholipid bilayer. I am showing how NAD+ combines with H+ to create NADPH. This is to further the concentration gradient, where there are more H+ inside the plumen. Now I'm showing 4 H+ traveling through the ATP synthase to create ATP. All of these products now head to the Calvin cycle. The second phase of photosynthesis is called the Calvin cycle. So at the beginning of the Calvin cycle, you have a 5-carbon chain of RUBP. So phase 1 of the Calvin cycle is called carbon fixation, where you add carbon dioxide to create a 3-carbon PGA. The next phase is called the reduction reactions. And phase 3 is called regeneration of RUBP so that the cycle can happen again and again. So first, carbon dioxide and water are added to the cycle. And the product of the cycle is a G3P, which is used to form glucose. And in the cycle, ATPs are involved. Another intermediate is the one shown, the 1,3-BPG3C. NADPH is also converted to 2 NAD+. Rubisco is the enzyme used in phase 1, which is the most abundant enzyme on Earth. Now I am showing that the cycle must happen 3 times to get to the desired products. So I multiply each amount to get the proper values. Next, I will talk about photorespiration in a C3 plant. Rubisco is involved again in photorespiration. So I'm going to redraw the Calvin cycle to show how it interacts, but while doing that, I will explain photorespiration for you. So photorespiration occurs when there are low levels of carbon dioxide in a plant, and this occurs usually on hot, dry days, which causes the leaves stomata to close. They close in order to prevent further water loss. And once the CO2 levels start to drop in the plant, Rubisco combines with oxygen, and since it has a greater affinity, the result is a production of 1,3-C PGA and 1,2-C phosphoglycolate. And this furthermore leads to carbon dioxide forming. And this process is similar to humans using up fat stores in short food times. Next is C4 plants, and C4 plants try to reduce their need for photorespiration. So at the top there, I've drawn a mesophyll cell, and on the bottom, the bundle sheath cell. And there is the Calvin cycle in the mix of it all. So C4 plants are resistant to droughts, and they are able to reduce their photorespiration by pumping carbon dioxide into the bundle sheath. And it then outcompetes oxygen for Rubisco, and it yields a lot of ATP. Decarboxylation occurs when the malate, pictured, converts into pyruvate, and then becomes PEP, returning to the mesophyll cells with the help of ATP. So there's the mesophyll cell, there's the bundle sheath cell. Finally is cam plants, and these are plants like cacti and pineapples. So first I will show nighttime. At nighttime, the stomata are kept open. This allows for water to leave and carbon dioxide to enter. Once entered, it becomes oxyalloacetate combined with PEP to become malate, which is formed and kept in the vacuoles as malic acid. They're kept in the vacuoles so that they do not harm the rest of the cell with their acidity. Additionally, in the chloroplasts, starch is added to help convert carbon dioxide into the malate. So now for the daytime. They keep their stomata closed in the daytime so that the substances deflect and are not able to enter or exit. Daytime has almost the opposite happen as in the nighttime, where malic acid becomes malate again, loses its carbon dioxide, which is used to feed the carbon cycle, the Calvin cycle, and then the pyruvate is used to form triose. Triose then forms starch,